Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Today I want to show you how I prepare the lab simulation of restriction enzyme digestion. In this simulation, I will be performing a restriction digest. I will use restriction enzymes to cut the DNA strands of two plasmids, producing DNA fragments. Because we know the size of our plasmids and where the enzymes will cut the DNA, we can predict the length of the fragments that will be produced. We can also confirm the length of these fragments or determine the length of fragments produced from an unknown plasmid construct using gel electrophoresis. These DNA fragments can then be joined to make recombinant plasmids. So, what do I need to know before this laboratory starts? I will aim in this experiment is to produce two DNA fragments. One fragment contains a gene of interest, and the other fragment is a plasmid backbone that can be used to express proteins in bacterial cells. Ultimately, these DNA fragments can be joined together to create a recombinant plasmid for protein expression, a process referred to as gene cloning. The gene you aim to clone is called RFP. This gene is derived from a sea anemone, of a soft-bodied marine organism related to coral and jellyfish, and it produces a red fluorescent protein. So next for materials, the region that used in this lab simulation is buffer 2.5xb solution, PKAN-R plasmid, which is K solution, PARA plasmid A solution, restriction enzyme, and distilled water. So, in micropipette equipment used to measure small volumes. Next, empty tubes used to hold solution is A plus tube, A minus tube, K plus tube, and K minus tube. Other equipment is microcentrifuge, trash, floating tube rack, freezer, water bath, and ice bucket. So for the next section, this is the protocol of this lab simulation. First is set the temperature of the water bath. Second, set the volume of the P20 micropipette. Third, draw up the reaction buffer into the micropipette. Fourth, pipette the restriction buffer into the K plus tube. Five, pipette the restriction buffer into the K minus, A plus, and A minus tubes. Six, dispense the other solution into the K plus, K minus, A plus, and A minus tube. 7. Spin the solution tube. 8. Incubate the solution tubes at 37 degrees Celsius. And lastly, store the solution tubes in the freezer. So, can start the simulation. So first, set the temperature of the water bath. Okay, set the water bath temperature to 37 degrees Celsius. 37 degrees Celsius. Okay. So next, select the P20 micropipette and set the volume to 4 microliters. Next, attach a tip to the micropipette. Okay. Next, draw up the reaction buffer into the micropipette. First, move the restriction buffer to the empty tube rack and open it. Buffer into the empty tube rack and open it. Then draw up 4 microliters of restriction buffer. Next, pipette the restriction buffer into the K plus tube. First, open the K plus tube. Next, dispense restriction buffer into K plus tube. Okay, then eject the tip into the trash to prevent contamination. Okay, next, pipette the restriction buffer into the K minus, A plus, and A minus tubes. First, dispense 4 microliters of restriction buffer into the K minus tube. Okay, first attach the tip, then dispense 4 microliters of restriction buffer. Okay, then dispense into the K minus tube. Okay, then then eject the tip as usual. Next, dispense 4 microliters of restriction buffer into the A plus tube. So eject a tip, add a tip, then dispense buffer to the A plus tube. Eject a tip. Okay, next dispense 4 microliters of restriction buffer into the A minus tube. Okay, eject a tip. Next, dispense the other solution into the K plus, K minus, A plus, and A minus tubes. First, dispense 4 microliters of pKa into the K plus tube. pKa, eject a tip, then dispense into the K plus tube. Okay, eject a tip. Next, dispense 4 microliters of pKnR into the K minus tube. Okay, eject a tip. Next, dispense 4 microliters of PARA into the A plus tube. Okay. Then eject a tip. Next, dispense 4 microliters of PARA into the A minus tube. Eject a tip. And dispense to the A minus tube. Okay. Eject a tip to prevent contamination. Next, Change the volume of the P20 micropipette to 2 microliters. Okay. Next, dispense 2 microliters of restrictions enzymes into the K plus tube. Into the K plus tube. Okay, eject the tip. Then dispense 2 microliters of restriction enzymes into the A plus tube. Okay, add a new tip. Then dispense to the A plus tube. Okay. Eject the tip to prevent contamination and dispense 2 microliters of distilled water into the A minus tube. To A minus tube. Alright. Then eject a tip. Okay, so next, dispense 2 microliters of distilled water into the K minus tube. So as usual, add a new tip. Next, dispense distilled water into the K minus tube. Alright, done. So eject a tip. So next, spin the solution tubes. First, open the microcentrifuge, then place the solution tubes into the microcentrifuge. So, place the solution tube, then check the balancing, make sure balance. Okay, check balancing. So the centrifuge looks balanced and ready. Okay, close. Okay, set the microcentrifuge to spin for 10 seconds. Okay, start. Then remove the solution tubes. So drop the tubes into the rack to remove them from the centrifuge. Okay, done. So next, incubate the solution tubes at 37 degrees Celsius. Open the water bath. Insert the floating solution tube rack into the water bath. Okay, next set the water bath timer for 1 hour. Okay, set for 1 hour. Okay. Okay, wait for 1 hour. Okay, done. So next, store the solution tubes in the freezer. First, open the freezer. Next, place the samples in the freezer. So first, insert the floating tube right into the freezer. Okay. Then, place the samples in the freezer. Right. Okay, close. And that's all. This is the results. So that's all for the lab simulation of restriction enzymes digest. Thank you.